Okay. We are going to start talking about the regulation of gene expression, which involves a lot of pathways and operons. And so I'm going to switch back just for a couple lessons to the kind of old school way of doing this, just to make sure that everybody has everything that they need. Also, to slow me down because I feel like we kind of flew through transcription and translation. So we're going to kind of take a step back. Specifically, today we are talking about the regulation of gene expression. Regulation of gene expression. Boop. Okay. And this will kind of be most, if not all, of next week as well, especially because we're not going to get through all of this today. But we are going to start with the bacteria kind of side of things and how bacteria regulate gene expression. And they do so through regulating transcription. So, bacteria respond to their environment by regulating transcription. Bacterial response via transcription. Whoop. Regulation. And this kind of all boils down to the construct of an operon. We will be talking about two different operons for this lesson, so today and tomorrow. The trip operon and the lac operon. These two are very, very, very common when you are learning genetics. So, we are going to start with the trip operon. Our big example starting off is the trip operon. And I will tell you exactly what an operon is after we get into why we need it. So, tryptophan is an amino acid that bacteria use to build proteins, like you would use any amino acid. But tryptophan is synthesized from a precursor molecule in a 3-step pathway. So, tryptophan is synthesized from a precursor in a 3-step pathway. So, before we can get into the genetics of this, we are going to talk about the actual metabolic pathway. So, we are going to draw out the tryptophan metabolic pathway. So, we start out with our precursor molecule. That precursor molecule is transformed into an intermediate. And that is taken place, or that is catalyzed by enzyme 1. I said there are 3 steps. So intermediate 1 is transformed into intermediate 2 by something we are going to call enzyme 2, because I'm not going to make you memorize enzyme names. And finally, intermediate 2 is synthesized into tryptophan by enzyme 3. And while it is important for us to know how the metabolic pathway works, we want to know about the genetics. So, enzyme 1 is coded for by genes TRPE and TRPD. TRPE. Both of these code for enzyme 1. So its quaternary structure has 2 polypeptide chains, right? Because the polypeptide chain coded by TRPE gene and the TRPD gene come together to make enzyme 1. TRPC codes for enzyme 2. And TRPB and TRPA code for enzyme 3. Now, tryptophan can regulate gene expression. Regulate gene expression. Go ahead and put like a negative sign there because it is a negative regulation. So tryptophan can control all of these genes. I want to talk about how. That's what the whole goal is today. There is also, right here, negative feedback inhibition. Which we won't get super into today, but we have talked about feedback inhibition in the past when we talked about enzymes at the very beginning of the year. So this pathway is what we are working on controlling. And we are going to control this pathway, therefore tryptophan synthesis, through regulating how these genes are transcribed. Almost all regulation of responses to things, whether that be low levels of tryptophan or high levels of glucose, most of all of bacterial responses to that are through transcription regulation. Eukaryotes do it a little bit differently. They do this, but not on such a grand scale. So, now we have our background information. So, we have one promoter for all 5 of those TRP genes, TRP A, B, C, D, E, right? We have one promoter for all 5. This is our transcription unit. Remember, we talked about all of the genes that one promoter codes for. That's our transcription unit. All 5 genes are transcribed at one time, which means we make the corrections for enzymes 1, 2, and 3 all at once. Grouping genes like this is advantageous because we can have an on-off switch to control the whole cluster. So grouping genes is advantageous that's wrong. Oh well. Advantageous because we can control all transcription with one on-off switch. This on-off switch is called an operator. Lots of different genes have operators. Operators are positioned within the promoter or between the promoter and genes. Within the promoter or between the promoter and genes. And what they do, the whole point of an operator, is to control access of RNA polymerase to genes. Operators control the access RNA polymerase has to the genes. Controls RNA polymerase access to genes. If RNA polymerase cannot bind to a gene, RNA polymerase cannot transcribe. Therefore, by preventing RNA polymerase to bind, we don't make the mRNA to make a protein, therefore we do not make the protein. So, now we can get to what an operon is. An operon is the segment of DNA that is the operator, the promoter, and all of the genes. Operator, promoter, and genes. So it's that big long segment of DNA. So when we talk about the lac operon, we are talking about the whole segment of DNA that is the operator, the promoter, and all of our genes, A, B, C, D, E. That's the trip operon. So, how does all of this work? The trip operon can be switched off by a repressor protein. Trip operon can be switched off by a repressor protein. Repressors bind to operators and block polymerase. That's all they do. They bind to our operators and they don't let anything else in. That is all repressors. So a repressor binds to operators thought I could do it, and blocks RNA polymerase. Okay. The repressors are proteins, right? Because the repressor is a protein, it needs to be coded for by a gene. So a repressor protein is encoded by something called a regulatory gene. Repressor proteins are encoded by regulatory genes. Regulatory genes have their own promoter. Regulatory genes have their own promoters. And are expressed constantly at a low rate. So, the synthesis of these repressor proteins is always going on. But that doesn't mean that the trip operon is constantly repressed. That is because of two things. One, the binding of repressor proteins is reversible. So a repressor protein can bind to and stop a sequence from being transcribed, but it could also unbind and let transcription happen. So the binding of repressors is reversible. The binding of repressors is reversible. But also, specifically the trip repressor is allosteric. If you remember that way back at the beginning of the year when we talked about enzymes, allosteric means that there is an active and an inactive form, depending on if something is bound to it. So the trip repressor is allosteric, which means there is an active and inactive form. When it is synthesized, it is inactive. So its default state is inactive. So even though there is the repressor protein constantly being transcribed and translated, and there's constantly repressors in the nucleus, the trip operon is still going to function because that repressor is inactive. It is activated by tryptophan. Which makes sense, right? If there is tryptophan, if we have tryptophan, we don't need to make more. So the tryptophan binds to the repressor, activates it, and shuts off transcription. Because tryptophan has to bind to the repressor to make it active, this makes tryptophan a co-repressor. Which makes tryptophan a co-repressor. Co-repressors are any small molecule that cooperates with a repressor protein. A corepressor is any small molecule that cooperates with a repressor to switch off an operon. So, I think we have just enough time to draw this out. So, we've got a sequence of DNA. Start that out. We have a promoter. And what we are going to call trip R. This is our regulatory gene. It will get transcribed into mRNA, which will then make our inactive repressor. That's our regulatory gene. We have another promoter. We have our operator. And we have all of our trip genes. Trip E, trip D, trip B. Whoop, no, that's C. C, B, A. Way more DNA. All of this right here is the trip operon. So, if our inactive repressor, we'll say it's shaped like that. Let's say that inactive repressor combines with a trip molecule. This gives us our active repressor. When it is active, it will repress the synthesis of this. This repressor will bind to our operator and stop RNA polymerase from going through and making the enzymes that will make tryptophan. How we doing? All right. So that is one example of an operon and how we use transcription to regulate. Tomorrow, we will talk about the lac operon and how we synthesize lactose, or not we, but E. coli synthesize lactose. And then we might be able to start eukaryotic transcription, or not transcription, regulation. I don't know yet. We're still relatively at the beginning. Right. Have a wonderful day, y'all. I will see you tomorrow.